All right, I want to make sure that we have plenty of time for Q&A at the end of this session. So I'm going to go ahead and kick us off. So hello, welcome everyone to our RRP hosted webinar series. For those of you who I do not know yet, I'm Katherine Erickson, the Executive Director of the RUNX1 Research Program. For those who don't know the RUNX1 Research Program, we are a nonprofit organization dedicated to supporting patients with RUNX1 familial platelet disorder, which many of you know has a high predisposition to blood cancers. How do we support patients? Well, we do it through funding research, building and supporting our patient community, and of course, facilitating collaboration through mechanisms like this across our healthcare ecosystem, all with the goal of discovering treatment interventions that would prevent blood cancer in RUNX1 patients. I want to acknowledge our wonderful grant co-funding partners, Alex's Lemonade Stand Foundation, the Leukemia Lymphoma Society, the Mark Foundation for Cancer Research, as well as Evans MDS. Without them, we would not have been able to commit over $7 million in research funds over the past 5 years. We started these research webinars last year during COVID, and we found them to be a great way to keep the RUNX1 research community connected and marching forward. So you'll see that we plan on continuing to host these well into the future. And I just want to make a plea that if you are doing any RUNX1 FPD relevant research and are interested in presenting your research findings on one of our webinars, please reach out. I would love to hear from you. So before I introduce our speakers today, I'd like to remind everyone of the importance of the ongoing NIH RUNX1 FPD natural history study. Our two speakers today are investigators who are heavily involved in the study. And it's, as many of you know, it's one of a kind. It's the first of its kind, I should say, tracking in great high definition detail RUNX1 FPD disease over time across a very large cohort of patients and families. And we are especially excited about this particular study because the findings from this study have the potential to reveal drivers of cancer development and molecular markers that predict cancer onset. We at RRP want to thank all of the patients who have generously participated and contributed to the study. And of course, we also thank all of the investigators involved, most especially the PI, Dr. Paul Liu, and the medical director, Dr. Leah Cunningham. If you're a patient listening today or a clinician who cares for patients, please do consider contributing to the study. It is open and enrolling, and as you'll hear today, we are learning so much. Okay, without further ado, I'd like to introduce our first speaker, Dr. Keith Sacco, who is a clinical fellow at the National Institute of Allergy and Infectious Disease. He is going to be speaking to us about sex differences in allergic and immunologic disorders in germline RUNX1 deficiency. Take it away. Oh, actually, I wanted to make a quick announcement. If you have any questions, please do submit your questions in the Q&A icon, and try to refrain from asking questions through the chat. But of course, if you want to comment or make any comments, that would be the icon to use, the chat function. But questions in the Q&A icon, that way we can easily facilitate a Q&A session at the very end. So with that, I'll turn it over to you, Keith.
Thanks, Dr. Erickson, and thanks to the RUNX1 Research Program, Dr. Cunningham, Dr. Liu, who initially invited us to be part of this study. I'm a clinical fellow working in the lab of Dr. Guerrero at the NIH. And we initially got interested in germline RUNX1 deficiency because we do know that RUNX1 is involved in lineage maturation of lymphocytes, also the innate arm of the immune system. And ways by which the immune system can go wrong, I mean, there are a number of ways. 100 years ago, we would think that the immune system protects us from infection, but we also know that the immune system protects us from developing cancer. The immune system can also go wrong by reacting unnecessarily either against self, which would be autoimmunity, or excessively in forms of inflammation that could be allergic inflammation or non-allergic. So from our standpoint of immunology, we wanted to figure out whether lack of RUNX1 activity predisposes to allergic disorders, and if so, whether these are harder to treat, and whether there's a mechanistic basis from an inflammatory standpoint to support that. Also, we wanted to see if there are changes in immune regulation, particularly pertaining to broad inflammation and autoimmunity or susceptibility to infection. But as Dr. Erickson pointed out, the overarching question is whether these changes in the immune system predict the onset or severity of malignancy. So when we look at the literature, what we know in terms of immune responses when we look at RUNX1 mutations, Th2 inflammation is the predominant form of inflammation with classic allergic disorders. And we do see from literature that the wild-type RUNX1, when RUNX1 is working properly, it represses transcription factor GATA3, which is important to promote allergic inflammation. And this is actually more so when there is overexpression of RUNX1. However, when there is a dominant negative form of RUNX1, this was an experiment in mice. This led to increased allergic inflammation, predominantly seeing cytokines IL-4, IL-5, and IL-10 following stimulation of T cells. Subsequently, one form of inflammation we are interested in is what we call Th17 inflammation. This is the type of inflammation that we tend to see in Crohn's disease and psoriasis, those forms of autoinflammatory disorders. Also, Th17 inflammation has been documented in a few autoimmune disorders. And we do know that RUNX1 promotes the transcription factor ROR gamma T, which is important for this type of inflammation. And this paper back from the Lab of Clinical Immunology at the NIH had shown that RUNX1 is very important for producing IL-17, as you can see here in this graph, while with the dominant negative mutation in RUNX1, there was decreased production of IL-17. Moreover, a subset of cells we're interested in when it comes to autoimmunity and cancer predisposition, these cells are the T regulatory cells, which are very important for immune tolerance, as well as preventing allergic inflammation. And we do know that RUNX1 induces the expression of the transcription factor FOXP3, which is needed for the development and function of these T regulatory cells, together with Treg associated genes. And in experiments whereby RUNX1 was silenced with silencing RNA, there was decreased expression of this FOXP3 gene. This was also seen with the isoform RUNX3 and was more pronounced with both silencing of RUNX1 and RUNX3. This was an experiment done in human CD4 T cells. When we do see experiments in mice, cells that lacked RUNX1 in murine Tregs, these mice developed lymphoproliferation, autoimmune gastritis together with their corresponding autoantibodies, and hyperproduction of IgE. So there were features of both autoimmunity and increased IgE, suggesting that there is increased IL-4 signaling, suggestive of allergic inflammation. And you can see here in this graph, there's increased IgE when there was deletion of both RUNX1 genes, and this did not happen with deletion of RUNX3. There's an increase in IgE, but IgG levels were not affected. Also, deletion of RUNX1 led to increased antiparietal cell antibodies, which even in clinical medicine are associated with autoimmune gastritis, also known as pernicious anemia. And what they also did in this experiment is that they deleted the gene for core binding factor beta, which is, as a lot of you know, binds to the RUNT homology domain of the RUNX1 protein. And in these mice, the T regulatory cells had less suppressive activity when compared to the control mice. So given these observations, our hypothesis was that in patients that lack RUNX1 mutations, there would be increased Th2, also known as allergic inflammation. We were not sure in terms of Th17 responses. We did see that RUNX1 promotes Th17 responses. So we wanted to keep an open mind in terms of whether we observed Th17 inflammatory disorders pertaining to what the gene protein function would lead to. And we also hypothesized that there would be increased autoimmunity when RUNX1 activity was lacking. So we started our protocol by evaluating all these patients sequentially when they arrived in clinic. We did a few of these via telemedicine last year, and we wanted to characterize whether there was any particular striking immunologic phenotype pertaining to allergy, autoimmunity, severe and or recurrent infection. We also obtained clinical labs to evaluate the immune system, looking at mast cells by measuring the serum tryptase, looking at white count differential, quantitative immunoglobulins. We're also now looking at a screening autoantibody panel and performing blood tests to food and environmental allergens by looking at their IgE to see whether patients are sensitized. We are also now doing skin testing in the clinic when the patients visit. In terms of our lab research plan, we are currently immunophenotyping these patients and using the peripheral blood mononuclear cells to see what kind of inflammation the lineages of the immune system have, particularly looking at cytokine signaling. We are focusing to get age and sex-matched patients as our controls. And we are also excluding patients with clinical evidence of myelodysplasia and a history of malignancy to see, to prevent confounding the data. We're also performing naive T-cell differentiation assays. As you know, it's pretty hard as for human data to be, it is pretty heterogeneous. And we will also be performing Treg suppression assays. So currently we have evaluated 46 patients, half of them are female, and all of these patients have confirmed RUNX1 mutations. The median age is 39, the range of age is from 2 years to 74 years. The median age of females is a bit higher than males, being 42, and the males have a median age of 34 years. This is a protein showing the peptide structure of RUNX1, particularly highlighting the RUNX homology domain here in dark orange and the transactivation domain in green. The RUNX homology domain binds to DNA and core binding factor, and transactivation domain is important for activating the protein and interacting with other transcription factors and downstream proteins. And we have 16 unique variants in our cohort. A lot of the patients were in families, and we're annotating the type of variants by color code. Of note, one patient had a deletion in exon 3 to 5, for which the patient has loss of function in other genes. And this is probably the initial observation we made that was pretty striking. In orange bars, you can see the general prevalence of allergic disorders as reported in the United States. And in blue, this is the prevalence that we have in our cohort. As you can see, quite strikingly, the prevalence of allergic rhinitis, conjunctivitis, physician-diagnosed eczema, asthma, food pollen syndrome, and food allergy is markedly increased when compared to the background population. The proportion of classic IgE food allergy is likely lower, partially because our cohort may be older, and we do know that the prevalence of food allergy in the general US population is rising. We then wanted to look at whether there are sex differences within this cohort. And what is interesting to note is that when it comes to food-associated allergic inflammation, there is a marked skewing towards males, as is observed in the background US population. Same goes for eczema, there's a trend for males, and conjunctivitis, but not so for allergic rhinitis. Females also have increased prevalence of a history of physician-diagnosed asthma. When we look at allergic rhinitis and conjunctivitis, we have now tested either via skin tests or serum tests for environmental allergens. We've tested 34 patients. Of these 34, 30 had allergic rhinitis with or without conjunctivitis, and 21 of them had positive testing, be it on skin testing and or serum testing. This reflects the general rate of positivity we see in the background population. If these 5 patients have received subcutaneous allergen immunotherapy, also known as allergy shots, but none were on shots at the time of our evaluation, in terms of severity, most of them were controlled with either no sprays, intranasal glucocorticoids, or antihistamines, or oral antihistamines. And when we looked at sensitivity, as you can see in this heat map, each column represents one individual patient. A lot of the patients who were positive on testing were polysensitized. As you can see in this one patient being sensitized to grasses, dog dander, cat dander, and trees. When it comes to eczema, half the population in our cohort reported a pruritic erythematous rash. However, only 18 of these 23 that reported a positive history were formally diagnosed with eczema by a physician. When it comes to the skin inflammation, severity of eczema is assessed by looking at possible infections such as Staphylococcus molluscum or eczema herpeticum, but no patient had eczema herpeticum in our cohort. One patient had molluscum contagiosum, leading to mild residual scarring. And none of our patients have been on systemic immunomodulators or biologics such as dupilumab to treat their eczema. None of them actually had to be admitted to the hospital for an eczema flare. And most of them were treated with daily topical emollient, and their eczema resolved by late adolescence. When we look at asthma, 11 out of 42 patients we have evaluated had a physician diagnosis of asthma. It's important to note that anecdotally, as we're reviewing spirometry and lung volumes, the lung volumes are rather underwhelming and are not necessarily congruent with a classification of mild, moderate, or severe asthma. But we do have 4 patients who have a diagnosis of mild persistent asthma on daily inhaled corticosteroids, of which 2 patients are also taking the leukotriene receptor antagonist montelukast. We do have 1 patient who has severe persistent asthma that also takes the anti-IgE mepolizumab biologic. On top of that, 4 patients have had childhood asthma, which is now resolved, and 2 patients have exercise-induced asthma. When it comes to food allergy, predominantly, we do see this in males, as I've mentioned before. Of the 4 patients that have a classic history of food allergy, i.e. vomiting, hives on eating the same food, or anaphylaxis, we have 3 patients that are males. They have a food allergy to peanut, banana, and egg. 1 patient who has an allergy to flounder, fish, is female. 2 patients with eosinophilic esophagitis have biopsy-proven eosinophilic esophagitis, of which 1 patient has now had the inflammation which resolved on dairy elimination. And we have also observed 4 patients having food pollen syndrome, which is a form of allergy where patients experience itching, mouth itching or throat itching upon eating these foods mentioned. And this occurs usually due to cross-reactivity with environmental allergen pollens. And all these patients had either positive birch and or ragweed that cross-reacts with these foods. When we look at autoimmunity was largely present in females within our cohort. So while out of the 46 patients, 11 of them had a positive history for autoimmunity, 9 of these 11 were female. As you can see, there is a lot of heterogeneity in terms of diagnosis for autoimmunity. We do have 2 patients who have alopecia universalis, but both patients are siblings, so it's unknown whether there's another gene that is contributing to this. We also do have 1 patient not shown here who has the exon 3 to 5 deletion, who has a possible autoimmune myopathy in the setting of positive acetylcholine receptor antibody. If we look at labs, what is striking, as you can see, patients with Germline RUNX1 deficiency have lower platelet counts as compared to the general population. The reference ranges, the outside limit of normal, are grayed out, but there is nothing that is extremely striking when we look at the white cell differential, looking at absolute neutrophil counts and absolute lymphocyte counts. Serum tryptase, which is a marker of mast cell activation, is within reference range, as are the medial levels of serum immunoglobulin G, immunoglobulin A, and immunoglobulin M. Serum IgE, which alludes to allergic inflammation, is higher in males, and these males that have higher levels of IgE, they all have some form of eczema, and they are all younger than 20 years old. If we look at peripheral lymphocyte subsets, we don't see any huge differences in terms of total T cells, CD4 T cells or CD8 T cells, as well as B cells. We do see, however, that females, as compared to males, they tend to have a lower median number of natural killer cells being important for anti-tumor immunity and antiviral immunity. Of note, we do have a couple of patients who have reported either a severe meningitis in childhood that hasn't been verified as being a viral meningitis, and one patient that has had recurrent herpes infection but wasn't on prophylactic antibiotics, and their NK cells were below the lower limit of normal. So in essence, I hope I've shown you, we do see trends that autoimmunity is more predominant in females within this cohort, while allergy, particularly food allergy, is more prominent in males, albeit we do have a younger cohort of males when compared to females. Our plan is to look at specific T cell subsets going forward, and looking at B cell subsets, perform our Treg suppression assays, as well as try to figure out whether genotype-phenotype correlations exist, and whether any form of these immune regulatory disorders predispose to development of malignancy. This work takes a village. I really appreciate being given this opportunity, and thank all the collaborators as well as the patients who've agreed to participate. And I'll be happy to take any questions after Dr. Calvo's talk. Thank you.
Great, thank you so much, Dr. Sacco. I'm gonna now turn it over to Dr. Kathy Calvo, who is a senior research physician in hematopathology at the NIH Clinical Center, and she will present on the spectrum of bone marrow features in RUNX1 familial platelet disorder. We can't hear you. Kathy, are you?
Got it, thank you. So the goal of the hematopathology part of our study at NIH is really to characterize the bone marrow features and function in baseline RUNX1 patients with thrombocytopenia, and also in patients who undergo disease transformation, to identify bone marrow and molecular features that are associated with or predictive of transformation. So what I'm going to, hold on a second. What I'm going to show you or cover in this talk are peripheral blood indices, bone marrow morphology, including cellularity, megakaryocyte maturation, erythroid myeloid maturation, malignant transformation, and other findings that we've had. Additionally, I'll go through bone marrow flow cytometry maturation patterns, cytogenetics, and NGS mutation panel analyses. So if we look at our cohort at NIH, 87% of patients that we've looked at in the hematology section have thrombocytopenia. Interestingly, we have several patients who have normal platelet counts. However, my colleague, Dr. Alina Dulau, has been doing platelet aggregation studies on the RUNX1 cohort, and all of these patients, even with normal platelet counts, have abnormal platelet function. When we look at mean platelet volume, which is basically a measure of the size of the platelets, we really see a number of patients who have higher platelet volume and a number with lower. So it really is skewed or scattered across with a large distribution. Looking at hemoglobin levels, so hemoglobin reference ranges vary depending on whether the patient is a male or female and the age range. So everybody that is indicated with a red dot has an abnormal value. So about 26% of our patients had low hemoglobin levels for age. And then when we look at the mean cell volume of red cells, the majority of patients with anemia had a macrocytic anemia. As Keith mentioned earlier, when we look essentially at neutrophils, lymphocytes, and monocytes, the majority of patients really fall within the normal range for these particular lineages in the peripheral blood. However, 30% of our patients actually have increased eosinophils, which could be related to the allergic findings that Keith, some of them that Keith went over. So to date, we've had bone marrow biopsies on 39 patients with germline RUNX1 patients, and I'm going to go over these findings. I'm not going to include the patients who've had hematopoietic stem cell transplantation. We are following several patients after transplant as well. So in the cohort that I'm going to review, we have 24 adults, 7 are male, 17 are female, and 15 pediatric or adolescent patients. 10 were male and 5 were female. So overall, looking at the whole cohort, the majority of patients, 46% had hypocellular bone marrows for age, and about 38% had normal cellular marrows. So when we look at the pediatric versus the adult populations, the majority of adults had normal cellular marrow. However, the majority of our pediatric patients had hypocellular bone marrow for age. We only had 6 patients with hypercellular marrows, and 5 out of those 6, their disease was associated with leukemia. Looking at the megakaryocyte numbers, the majority of patients actually have normal megakaryocyte numbers in the bone marrow, and about 30% have increased megakaryocytes. And then we have a smaller number, about 13% have decreased megakaryocytes. And a number of these cases were the leukemia patients. In terms of megakaryocyte morphology, it's very important to note that about 72% of our patients had abnormal or atypical megakaryocyte morphology, and I'll show you that in a bit. About 8% had dysplastic megakaryocytes. And really only about 10% had normal megakaryocytes. Looking at myeloid and erythroid lineages, the majority of patients had normal myeloid and erythroid lineages. Only 10% showed dysplasia in myeloid and erythroid lineages. So overall, about 21% of the patients who've had bone marrow evaluation have had evidence of malignant transformation. And in adults, we've had 2 cases of MDS with ring sideroblasts, 1 chronic myelomonocytic leukemia, 1 acute myeloid leukemia, and 1 patient with smoldering myeloma. In the pediatric or adolescent range, we've had 1 case with B-ALL, 1 with T-ALL, and 1 with acute myeloid leukemia. So other findings that we've had, we have 1 patient who has monoclonal B-cell lymphocytosis, another patient with MGUS, which is monoclonal gammopathy of undetermined significance, which is a precursor lesion to smoldering myeloma or myeloma. We've had 5 patients with absent B-cell precursors and 15 patients with increased eosinophils in the bone marrow. Of our 3 patients so far that we've been following who've had transplant, 2 out of the 3 had marrow that was hypocellular for age with normal trilineage hematopoiesis. So now I'm going to go into our bone marrow flow cytometry analysis of patients. And in this part of the study, we actually had 16 age-matched adult healthy controls, 20 adult RUNX1 patients, 9 pediatric or adolescent patients. defined as being less than 18 years. So in this particular part of it, we've excluded the leukemia and CMML case, really to get a sense of what the baseline RUNX1 bone marrow looks like in terms of cell lineages by flow cytometry. So you can see here in terms of the age distribution, there's no significant difference between the healthy control adults and the RUNX1 adults. So the median age of the RUNX1 adults was 42, the range was 27 to 72. Pediatric population, median age was 10, range of 3 to 17. And these are our post-transplant patients. So looking at bone marrow monocytes, the RUNX1 adult population had slightly higher, significantly higher numbers of bone marrow monocytes in comparison to the healthy controls. When we looked at eosinophils, we could see a significantly higher number of eosinophils overall in the bone marrow in the RUNX1 patients in comparison to the healthy controls. And when we looked at lymphocytes, we saw higher overall percentages of lymphocytes in the bone marrow of the RUNX1 patients in comparison to the healthy controls. Looking at lymphocyte subsets in the bone marrow, overall, the RUNX1 patients had higher numbers of T cells. However, that was not statistically significant. So this was just a trend that we saw. When we looked at the CD4 to 8 ratio, overall, the RUNX1 patients had inverted CD4 to 8 ratios. However, it was not statistically significantly different from the controls. Looking at mature B cells, really, we did not see any significant difference in the RUNX1 patients in comparison to controls. And overall, looking at precursor B cells, the difference was not significant. However, we always take a careful look at precursor B cells when we're evaluating patients with potential MDS, because one of the findings that we commonly see in MDS is loss of early B cell precursors. So in about 4 or 5 patients, we had very low to absent precursor B cells. Looking at NK cells, overall, there were significantly decreased numbers of NK cells in the bone marrow in the RUNX1 group in comparison to the controls. And this is, as Keith showed in the peripheral blood, he also found this finding primarily in the female RUNX1 patients. So CD34 is a marker for hematopoietic stem cells. It also marks blasts in the bone marrow, and we saw no significant difference in the numbers of CD34 positive cells between the healthy controls and the RUNX1 adults. As you can see here, the pediatric patients have much higher CD34 positive cells, which is normal and often correlates with relatively increased numbers of B cell precursors that children have. So there's been a lot of interest in CD123 based on some research studies, and we looked at CD34 positive, CD123 positive cells in the bone marrow of RUNX1 patients, and they were not significantly increased in comparison to healthy controls. And this was for our baseline RUNX1 patients. However, one of the patients who developed CMML actually did have an increase in the CD123 positive CD34 population. When we looked at dendritic cells in the bone marrow, we did not see a significant increase or decrease in the RUNX1 patients in comparison to controls. However, basophils were significantly decreased in the RUNX1 population in comparison to controls. So now I'll go over some of the morphologic findings. So normal megakaryocytes in the bone marrow undergo a process called endoreduplication, where there's DNA synthesis without mitosis, resulting in polyploidy. So this is a normal megakaryocyte, and it has multiple nuclear lobes, which are all connected. This is on a core biopsy, what it looks like, and this is on an aspirate smear. So in our RUNX1 patients, about 86% of patients have abnormal or atypical megakaryocytes. Many of them are very small, hypolobated with an eccentric nucleus. We have patients that also have megakaryocytes with separated nuclear lobes, as you can see here and here. These are core biopsy images, and this is an image on an aspirate smear. So this is actually a very important finding because, which has also been reported previously by others. Why is it important? Because in the general pathology and hematology community, these type of megakaryocytes would be considered to be dysplastic and may meet criteria in a normal setting for a diagnosis of MDS. However, it's really important for us to get the word out that RUNX1 patients have these abnormal appearing megakaryocytes at baseline, even without having MDS. So that really, in order to diagnose MDS in a RUNX1 patient, we need to have more than just atypical megakaryocytes. We need to have either increased blasts or multilineage dysplasia or cytogenetic abnormalities, because these are essentially normal for RUNX1 patients and are indicative of abnormal megakaryocyte maturation, which is associated with thrombocytopenia or platelet dysfunction. We've had 2 patients with clonal plasma cells. This is an example of a patient who had 10 to 15% plasma cells in the bone marrow, which is increased. And this is how the plasma cells look on the aspirate smear. So by flow cytometry analysis, we gate on cells that are bright for CD138 and CD38. These are our plasma cells. And what we found in this particular patient were these plasma cells were markedly abnormal and that they lacked CD19 expression, which is expressed on normal plasma cells. Additionally, when we looked at cytoplasmic kappa and lambda, kappa was markedly skewed, which was consistent with the clonal plasma cell population. Again, we've had about 5 patients with absent B-cell precursors. So this is a normal control. What we find, this is the lymphocyte gate, CD10 and CD20. Early B-cell precursors are bright for CD10, they're negative for CD20, and as they mature, they slowly downregulate CD10 to a moderate level and begin to slowly express CD20, and then these are our mature B-cells, which are negative for CD10. So we have a number of patients that have absent B-cell precursors. Again, this can be concerning for a number of reasons, that this may be either an early indication of potential B-cell immunodeficiency in the future, or this also can be associated with MDS, although these patients did not have MDS. About 30% of patients had increased eosinophils in the bone marrow. So now transformation to myeloid neoplasm or lymphoid neoplasm, CMML, AML, MDS, and acute leukemias. So these are images from a 58-year-old female with germline RUNX1 mutation and history of thrombocytopenia. Before coming to NIH, she had a prior diagnosis of MDS with 5q- on cytogenetic analysis. When she came to NIH, she had a markedly increased white count of almost 50,000. And we could see that she had circulating blasts and circulating immature monocytic precursors. Flow cytometry analysis showed about 8% myeloblasts that had abnormal expression of CD7, which we can see in dysplasia. So she had a marked leukocytosis with left shift. She had a left shifted monocytosis and circulating blasts. The blasts had a typical myeloid phenotype. They were positive for CD34, 13, HLA-DR, CD117, and CD33. So this was her bone marrow biopsy. So this is markedly hypercellular for her age. And we could see evidence of dysplastic megakaryocytes that were hypolobated. And another stain that we use by immunohistochemistry is CD61, which allows us to identify what are called micromegakaryocytes. These are always abnormal and they're megakaryocytes that are about the size of other myeloid cells in the bone marrow. But the most concerning thing was the CD34 immunostain. So typically, normal bone marrow should have less than 1 or 2% CD34 positive cells. So this was increased at 8 to 10%. This was her bone marrow aspirate smear, and we can see blasts. Blasts have a very high nucleus to cytoplasmic ratio and have what we call very fine chromatin. And additionally, we could see dysplastic megakaryocytes. This is a hypolobated megakaryocyte with an eccentric nucleus. And then here is a micromegakaryocyte. This is about the same size as other myeloid cells or blasts. And then she also had hypogranular myeloid cells. So this is consistent with dysplasia in the myeloid and the erythroid lineage and increased blasts. And the bone marrow flow cytometry showed markedly increased monocytes, so about 20% monocytes. Normal bone marrows usually have around 4%. The monocytes were left shifted, and approximately overall there were about 6% monoblasts that lacked expression of CD14. Again, she had about 7% myeloblasts with a typical myeloid phenotype. So this patient was diagnosed with chronic myelomonocytic leukemia with increased blasts and germline RUNX1 mutation. Interestingly, her cytogenetic analysis came back as normal. A somatic mutation panel identified the RUNX1 mutation with an additional NRAS mutation, a B-core mutation, and a PHF6 mutation. So this is a marrow from another patient who is a 42-year-old male with pancytopenia. Interestingly, this patient came to NIH for a different protocol to be transplanted for acute myeloid leukemia. And his bone marrow was packed, very markedly hypercellular. We could see very dysplastic megakaryocytes here with separated nuclear lobes. And this is the CD34 stain, which has a marked increase in blasts that were over 20%. So this patient was diagnosed with acute myeloid leukemia. Now, the interesting thing was that an NGS, a somatic NGS mutation panel was ordered, and the patient had a RUNX1 mutation with a variant allele of 50%. So this suggested that this patient might have a germline RUNX1 mutation, which was later confirmed on the patient and also in other family members. So this patient then joined the RUNX1 protocol. So again, he was diagnosed with acute myeloid leukemia, in this case with myelodysplasia-related changes and germline RUNX1 mutation. We also had an additional patient with acute myeloid leukemia. Here's an example from a 72-year-old female with a history of thrombocytopenia. When she came to NIH, her bone marrow was hypercellular for age. She had evidence of atypical megakaryocytes. This is the CD61 stain, which showed a subset of very small megakaryocytes consistent with micromegakaryocytes. Her aspirate smear actually looked fairly okay. The myeloid maturation looked good. Cells were well granulated and segmenting. The erythroid precursors looked okay. However, what was very abnormal was the iron stain. So she had ringed sideroblasts, which are always abnormal. Her cytogenetics came back normal, but she was diagnosed with MDS with ringed sideroblasts. We also had an additional patient with MDS and ringed sideroblasts. So one of our 17-year-old males was diagnosed with acute lymphoblastic leukemia. And interestingly, his molecular analysis showed an EBF1 PDGF beta fusion. So he had a Philadelphia-like BALL, which is a more aggressive form of acute lymphoblastic leukemia. We also have a 4-year-old male with a germline RUNX1 mutation who was diagnosed with T-cell lymphoblastic leukemia, and he had very complicated cytogenetic analysis. And his cells had cytoplasmic positivity for CD3. Surface CD3 was negative, CD5 was positive, and this was the phenotype of an earlier T-cell acute lymphoblastic leukemia. So we've done a research NGS panel on about 33 of the adult RUNX1 patients using a QIAGEN panel containing 141 genes. And in our first round of analysis, we've looked primarily at 42 of the genes that are most commonly mutated in myeloid malignancies. So on this panel, we identified 13 different RUNX1 variants in our patients, and you can see the distribution of mutations. In this particular panel, we were unable to identify the RUNX1 deletion, which is also important to note that many somatic mutation panels will not be able to pick up deletions. So out of these 33 patients, 24 of the patients, we only identified the RUNX1 mutation in, and they had no additional mutations that were identified. However, 8 of the patients, we did identify additional mutations. All 4 of the patients in this particular group that had myeloid neoplasms had acquired mutations in either NRAS, PHF6, BCOR, SF3B1. Both patients that had MDS with ring sideroblasts had SF3B1 mutations, which is not surprising because MDS with ring sideroblasts is associated with SF3B1 mutations. We also found mutations in JAK2 and EZH2, and you can see those 4 patients are 58-year-old female, 43-year-old female, 42-year-old male, and 72-year-old female. So out of the remaining 29 patients, only 4 had additional mutations that we detected. And the most common gene that was mutated was BCOR. We also found mutation in NRAS and TET2. One of our colleagues, Kai Yu, working with Paul Liu, has also detected DNMT3-1 mutations in a subset of patients. So summary points, as we know, RUNX1 patients typically present with lifelong thrombocytopenia. Patients are at high risk of transformation to myeloid malignancy and also to acute lymphoblastic leukemia, as we've seen. Important to note that baseline RUNX1 bone marrows show evidence of megakaryocytic atypia that can resemble dysplasia, even in patients without MDS. And that, so that patients, RUNX1 patients that have atypical megakaryocytes that look dysplastic in the setting of isolated thrombocytopenia, that finding really is not sufficient for a diagnosis of MDS in this population. That transformation to MDS or myeloid malignancy is associated with additional findings that may include additional cytopenias or pancytopenia, multi-lineage dysplasia involving either myeloid or erythroid lineages in addition to the megakaryocytes, ring sideroblasts, cytogenetic abnormalities, increased blasts, or additional mutations. However, the presence of additional somatic mutations does not necessarily indicate transformation. So at NIH, we're doing yearly bone marrow biopsies on patients with cytogenetic and molecular analyses in order to identify transformation early in the disease process, to identify patients that are at risk for transformation to MDS and leukemia, and to help decide when the best timing is for consideration of hematopoietic stem cell transplantation. So I'd like to thank my hematopathology colleagues, Dr. Alina Dulau, Raul Braylin, Nisha Patel, Irina Merrick, and Wei-Xing Wang, Megan Trick, and others in the clinical center. Of course, Dr. Paul Liu, Leah Cunningham, and Matthew McGarrian in NHGRI, who are leading the RUNX1 protocol at NIH, and all of our technologists. Thank you.
Great, thank you so much, Dr. Calvo. That was a beautiful presentation and very comprehensive. We have some questions, and but before, I'm going to be selfish and ask one of my questions, which is, were you able to do any correlative studies in that you described about 46% of patients had hypocellular marrow, and I'm curious whether any of those also had evidence of somatic mutations. You also mentioned the 14% that had BCOR, NRAS, or TET2. Do they then also have any sort of phenotypical changes or abnormal changes in the bone marrow?
So we haven't seen any specific correlation to date. That could change though as the study expands and we have more patients to look at. I mean, definitely the hypercellularity all of those patients had mutations and were associated with either MDS or leukemia. So we can definitely say that a hypercellular bone marrow is concerning. In terms of the normocellular and hypocellular, the jury is still out.
Got it. Thank you. All right, so here we go with some of the questions. I'll start off with the first one that I see in the list here. Are there any insights so far from the immune studies that would make patients cancer prone? This might be for you, Keith, that you think. Any hypotheses there?
That's a great question. So there are some trends we're seeing. I think I'm not in a position to kind of throw it out there. Hopefully by September, we'll have some more data to support it. But there are some trends of inflammation. They're not classic, Th2 or Th17 that we are seeing. That may lead to that.
Then another follow-up question, or a different question actually, but from the same person, Dr. Ponce. The macrocytic anemia is interesting. Were there any related family members? There, older age relationship, MDS, relationships to bone marrow analysis? I think that's for you, Dr. Calvo.
I'm sorry, can you repeat the question with regard to macrocytic anemia?
That it's an interesting finding, and were there any related family members? In other words, was one family having the same macrocytic anemia?
That's a really good question. We'll have to go back and look at that. I mean, macrocytic anemia is a finding that we often see in MDS, although it's not diagnostic of MDS in and of itself. We'll have to go back and look at that though.
What specific platelet function studies were performed in the cohort?
So Dr. Alina Dulau in our group has done platelet aggregation studies. One of the limitations of the studies that the patient has to have a platelet count of greater than 100. So we've only been able to do it in a subset of patients. And actually, she may be able to present the data at a future webinar. But all of the patients, we did have some patients that were initially we were evaluating with synonymous mutations, or synonymous variants, I should say. None of those patients had abnormalities. And as Paul would say, that we believe those were benign variants. So excluding the patients with potentially benign variants, all of the patients with pathogenic variants in RUNX1 had abnormal platelet aggregation studies. And I believe it was to collagen, and it was present, abnormal aggregation in response to collagen was present in nearly all patients. And also they didn't have the ATP release. So those are the primary. Also, I believe they're doing EM studies on platelets. I don't have those results. And Dr. Raul Braylin is doing EM studies on megakaryocytes in the bone marrow of RUNX1 patients. And maybe at a later date, he can go over some of those results.
Yeah, that would be great. Okay, the next question is for Dr. Sacco. I wonder if you have considered using family members without RUNX1 mutations as controls in your studies to control for many confounding genetic and environmental confounders in your results.
Yeah, great point. And we discussed this actually within our group. We actually do use these the family members, and it's very interesting to note there do seem to be differences. Of course, if we have a female family member, we'd have to match it with a RUNX1 female that would be from a different family. Also, when we look at our healthy volunteers, in terms of allergic inflammation, specifically, let's say, mentioning allergic rhinitis, they're pretty comparable in terms of prevalence. So our controls are not totally, non-allergic controls to see if there's actually a difference pertaining to, RUNX1 inflammation.
Are the sex differences consistent with the general population, or is that a RUNX1 phenomenon?
There is a trend, yep. So there is a trend. Allergies, food allergies tend to be more common in men, autoimmunity more common in females. We do see that. The magnitude seems to be greater. Again, we don't have. 40 for RUNX1 is a big population for the disorder, but still a small number, and so it's pretty hard to tease out. Even in terms of the patients who come in. Again, we're looking only on patients pre myelodysplastic syndrome or lymphoproliferation.
Great. Dr. Calvo, question for you. How old were the 4 patients with evidence of clonal hematopoiesis and no malignancies? And also curious if those patients happen to also have the lack or loss of precursor B cells.
That's a very good question. I don't have the answer to whether or not those were the specific patients that had the loss of precursor B cells, but we definitely should go back and look at that. Thank you for that suggestion. The ages of the patients that had the clonal hematopoiesis, I would have to go back, hold on, and look at my slide. The ages, 42-year-old male, 41-year-old female, 59-year-old female, 59-year-old male. So those were the ages of the patients who did not have malignancy, but who had clonal hematopoiesis.
Great. For you again, Dr. Calvo, was there any correlation of patients with megakaryocytes containing separated nuclei and the presence of secondary mutations found on NGS? Everyone's interested in these correlations.
Yeah, that's a great question. I can tell you that I know that a paper was submitted or published a couple of years ago, and actually there was even a presentation at one of the RUNX1 research meetings on megakaryocyte morphology, looking at megakaryocytes that had separated nuclear lobes, and that was associated with STAG2 mutations. We actually did not specifically see that in this cohort, although I can say for another disease that we look at, GATA2 deficiency, a number of those patients do have megakaryocytes with separated nuclear lobes and STAG2 mutations, but we haven't seen that in the RUNX1 cohort. We can go back, and not everybody has had the NGS analysis. Most patients have. We can go back and look at that, but it's not something that stood out.
Are there screening recommendations for young adults with AML to identify allelic and partial allelic RUNX1 deletions?
Are there screening, do you mean in the regular AML population or in, sorry, could you repeat the question?
It sounds like in young adults with AML.
Are they screening for RUNX1 mutations?
Yes.
We basically believe that any young person with AML, and at this point, many people would consider anybody under 40, if there are some researchers that would say under 50 that have AML, should be screened for germline mutations, yes. Is that being done? I think in most centers now, people are getting at least a myeloid somatic mutation panel, which would pick up a number of RUNX1 germline mutations or mutations in other genes such as GATA2 and KRD26, a whole list of genes that are mutated in patients with germline predisposition to myeloid malignancy. The problem is that many of the somatic panels really look at hotspots of the gene, and they may not pick up mutations that are not included on that panel. Additionally, even our panel was unable to pick up the patient that had the RUNX1 deletion. So in reality, it should be happening. And I even showed an example of one patient who had AML here. It was unknown that he had a germline RUNX1 mutation until he had the somatic panel and it was identified. And then the germline RUNX1 mutation was confirmed. So I think we're going to be catching more and more of these patients in the future. However, it's not perfect. probably whole exome sequencing is really needed to, and maybe even whole genome sequencing, you know, for patients that have mutations in intronic regions, to be able to identify everybody that has germline mutations in RUNX1 and other genes. This is sort of a long answer, but we're getting there, but we're not there yet.
Yeah, so there's really no specific recommendations that you're aware of to pick up the deletions at this point.
I think in many papers in the literature, it is being suggested. A lot of clinicians will say that, you know, well, who's going to pay for the testing? And so I think it is going to become standard of care probably in the near future, that any young person with acute myeloid leukemia gets gets a panel, a gene mutation panel.
Absolutely. So we are over time. I'm willing to stay online a little bit more. Are you both able to? There are still more questions. Okay. The next question is from Dr. Wenbin Zhao. Have you seen any cross-lineage marker expression in transformed cases?
You mean abnormal expression of markers by flow cytometry? I think that's what you're referring to. Yes, we have, and I think I showed there patients that have MDS. We've seen a number of them have abnormal expression, for example, of CD7, which is a T cell marker on myeloid cells, and abnormal loss of antigen expression. Yes, that's commonly seen in patients who develop malignancy. We haven't seen it specifically in patients who have not developed malignancy.
I have a question about the reduced B cell precursors and how you were saying these were concerning findings. I'm curious, are you as a clinical group changing the management of or surveillance of those patients?
No, I don't think that would meet. No, I mean, that's just, it's a finding that is sort of a soft finding. I don't think you would change management based on that finding. We would just want to monitor that patient, you know, over time.
So you wouldn't increase monitoring, I guess is the point I'm trying to,
not specifically for that. Now, if there were other findings, you know, if there were low B cells in the peripheral blood and additional findings, then maybe, but as an isolated finding, it would be something that we would just watch.
Do you have a comment?
To tag on to that, as Dr. Calvo said, so we've had a few patients who have low normal B cells, but no overt infection, and immunoglobulins were low normal. We don't start intravenous immunoglobulin, but we tend to trend it out, let's say, every 6 months instead of every year. But it hasn't seemed to be a problem so far.
There's another question around RUNX1 patients with variants of unknown significance, and whether, if those individuals were to have clonal hematopoiesis, would that be strong enough evidence to suggest that this was a disease-causing mutation?
I don't think I can answer that question. I don't think I could answer that question. I don't think we know. Maybe there are other people who've done enough studies to know.
Another question is, megakaryocytic ploidy abnormal in RUNX1 mutated patients. I think you covered this. Yeah, so and also Dr. Raul Braylon is doing ploidy studies on these patients. So yeah, I mean, you can just see morphologically all of the cells that have, most megakaryocytes, I believe, have up to 8 or 16 even nuclei by flow cytometry ploidy analysis. So a number of those small mononuclear megakaryocytes would be completely abnormal. I see a couple of questions from patients. So, have there been cases where platelets are a normal count in young children, and then the thrombocytopenia develops later? Do you have any evidence for that?
We don't, I mean, not specific. We haven't been following our cohort out long enough to answer that question. But in the future, that would be something that, you know, we would be able to answer or look into.
What I can add to that is that we do have a couple of patients who've had inflammation, let's say, and platelets rise with inflammation. So the platelets would be in the normal range once they get on treatment for that said inflammation, let's say, you know, colitis, then the platelets would come back to, you know, the low level we see in Germline RUNX1. So probably they can get up to a normal level, but it's a marker of
in response
to inflammation, right?
And another question from a patient is, should patients receive whole genome sequencing to fully identify any other mutations in addition to RUNX1? And does the NIH study currently plan on doing this on all patients who enrolled?
Sorry, I couldn't hear you that well.
Should patients aim to receive whole genome sequencing to fully identify any other mutations in addition to RUNX1? And will or does the study, the ongoing natural history study, plan on doing this on each patient?
Yeah, I believe that Paul Liu is doing those studies right now in his laboratory. That's my understanding.
I don't know if he's still online, but maybe I do believe that was my understanding as well, that everyone would get whole genome sequencing. All right, and then I have one question to cover here, which is, did any patient have, actually there's two more. Did any patient have autoimmune cytopenia?
Go ahead, Dr. Calvo.
You know, that's a really interesting question, because we've seen that a number of patients that have RUNX1 had prior diagnoses of, quote, ITP. So it's because ITP is actually a much more common cause of thrombocytopenia than RUNX1 FPD. So what we think is that there are a lot of patients who, if they don't know that if the RUNX1 mutation is not known and they get worked up for thrombocytopenia, they may receive a diagnosis of ITP when really what they have is RUNX1 FPD.
And then the final question is, for those with MDS ring cytoblasts, are you planning on doing NGS on the mitochondria?
I haven't. I'm not sure if Paul Liu's lab may be doing that study, so I'm not aware of that.
All right, I think we've gotten through all the questions. I really appreciate both of you, Dr. Calvo and Dr. Sacco, for being, for presenting and taking the time to put together such beautiful presentations and sharing the latest and greatest from the ongoing study. We really appreciate all that you do. So thank you very much, and thanks to all the attendees for the great questions. And hopefully we'll see you all soon again on another research webinar. Take care, everyone.